In this lecture we're going to see how antibacterials work by affecting protein synthesis. Let's start off with a strand of RNA. This is our messenger RNA, and we're going to work through the protein synthesis steps and figure out how we can mess it up with drugs. So here are 3, 4. So here are the triplets of codons. For example, here this is one triplet, and we're going to draw our RNA sat here, so that's the 30, this is the 50S, so this is the ribosome, and at the moment there we have a piece of tRNA no, that's all wrong too. Here we're going to look at how antibacterials work by affecting protein synthesis. So let's start with a strand of messenger RNA. We're going to work through the steps of protein synthesis and see how we can mess it up. So I'm drawing the triplets. These are the codons, the triplets, and we're going to have our RNA, sorry, our ribosome here. This is the ribosome, and we've got a tRNA reading off this triplet, and it's got its sequence of amino acids coming off here. So along comes another tRNA. Let's see if I can move this whole thing to the side. Here's another tRNA, and it's got its own amino acid attached to it. First thing that can happen here is that tetracyclines, for example, can interfere by competing with the tRNA for the site that it wants to land on, so it doesn't get to land. So that's one thing we can do. Let's get rid of the tetracyclines. So we've got tetracyclines, that's one way of attacking it. Then the next thing that happens is this codon here now lands. I think it's going to be easier if I just redraw it. This whole codon lands, attaches itself, sorry, not the codon, what am I talking about? The tRNA attaches itself to the next codon. This process of attachment can be interfered with by aminoglycosides, such as gentamicin. So that's another way we can get an attack. And then the next step in protein synthesis is the transpeptidation, which is where this string now gets attached to here, and drugs which block that are inhibitors of transpeptidation, such as chloramphenicol. The next thing that happens is that the whole transfer RNA kit moves one step to the left. Let's see if I can draw that. Maybe not. Maybe yes. Sort of. Anyway. And this step is interfered with by drugs like erythromycin. So then we've seen several steps at which these different drugs can act and stop protein synthesis from taking place. And if you can't make proteins, then as a bacterium, you're out of luck.